Hello, everyone who joined. Nice to see you all. Hello, Suraj.
Hello.
Yeah, nice to see you. Yeah, we are gonna wait for the rest of the people for a few minutes, and then I will start the presentation.
Okay.
Yeah. Hello, hello. As I said, we are gonna wait 2 more minutes, I think, before we start because not so many people here by now, and we should wait for them for a few minutes. Hello, Nishash.
Hello.
Yeah, nice to hear you. Yeah, I see some people from AIT. Oh, I want to ask you guys who joined already. Please rename yourself and add your affiliation, your university before your name, for us to understand who attend the meeting and who is not attending the meeting, who's absent as well. Hello, Kitut. Hello, Kitut. Nice to see you. I cannot hear you because you turn on off your voice. Hmm? Oh, yes, I see some people from Diana. I think we should wait for two more minutes for other people to join us.
Hello, Sergey.
Yeah, nice to hear
Are
you.
you, are you ready?
Yeah, yes, I am. But we're waiting for the rest of the people.
Yeah.
Yeah.
How many participants now?
I'm not sure, but I cannot see some people from. I see people from Diana. I see
Ah,
people from AIT from Thailand, and we should wait a little bit more. From maybe some people from
okay.
MMSU, from Vietnam. I cannot see any people from Vietnam.
Yeah, be proper to wait for a moment.
Yeah,
Okay.
but we will start very soon because we don't have so much time. Actually, we have 1.5 hours for the whole seminar, so we should start very soon.
Yep. Thank you.
Yeah.
Ah,
Hello, Professor Ha.
ah. How are you, Ha?
Yeah, how are you? Good, thank
Yeah.
you.
Hey,
you?
I'm.
Sorry, I come a little bit late.
No problem.
Okay.
Okay. What the season there in Vietnam?
The weather, you mean?
Yeah, the weather.
The summer is around.
Yeah.
So,
Oh yeah.
yeah, I think it's temperature more than 25
Oh,
Oh yeah.
and a lot of sunny. Yeah, I think the summer is around. Yeah.
well, I'm so
In
jealous.
Bali, it's okay now. It's a bit cloudy, and also but in many some areas like is Nusa Tenggara. You know that from the television that is about disaster of a cyclone.
Yeah.
Something like that. Yeah. Mm-hmm.
Okay.
In Moscow, it's cloudy and cold. Not so pleasant. In Moscow, we have a spring now, and it's okay. The summer will be very soon. So I think we can start our presentation because a lot of people here, some people from Vietnam, from AIT, from okay. Let's start. I will start a demonstration of PowerPoint. Okay. Can you see my PowerPoint? Is it okay? Professor Ha, you can see my PowerPoint. It's okay, right?
Yes.
Yeah.
I see.
Thank you, Kitu. Yeah, good. So, hello everyone. Nice to see you today at our second educational seminar in the project of adaptation and implementation of model molecular biology technologies in the practice of agricultural and wastewater treatment of ASEAN countries. So today's seminar will be devoted to DNA extraction techniques and the essence of polymerase chain reaction. These are the main methods which we will be using for detection of pathogens, for detection of microorganisms in the samples which we will collect later in the project. So we should understand how they work. Today we will go through some theoretical basics and then go through the detailed protocol of DNA extraction. I think I can demonstrate some equipment which we will use to extract DNA because I have this equipment with me, and maybe I will do the same as modern YouTubers do unpacking thing. You see, I have a box with DNA extraction kit, and I will show you what's inside, and we will discuss how it works. And then we will discuss polymerase chain reaction. This is very important topic. Today we will, I think, we will only start to learn how to do it because during our project we will not be able to perform polymerase chain reaction in each country, in local university, where you will work. We will extract DNA on site in local universities in each country, but then DNA should be transported to Moscow, and here we will perform polymerase chain reaction and maybe DNA sequencing as well. So we will not go through the detailed protocol of polymerase chain reaction today. It's very important and it deserves a separate lecture. Maybe we will discuss it later. Today we will only discuss how it works in general, and we will discuss the general workflow. Okay, but before we start to speak about this like molecular things, we should remember how bacterial cell looks what bacterial cell looks like. And then because we are going to work with bacteria and we are going to work with DNA of bacteria, we should think how it should be extracted and what we should do for it. On the slide, you can see bacterial cell anatomy, right? And the first thing I want to say is that we should understand that bacteria it is the whole organisms in one cell, right? Bacteria has nuclear cell wall, yes, which protects the cell from outside dangers and helps the cell to keep it warm. Bacteria cell inside the cell wall, we have cytoplasm of bacteria. This cytoplasm is the place where all processes are going. So here a lot of different proteins and all structures which needed for bacteria to live is here. For example, the very important structure, very important organelle is ribosome. Ribosome, we will discuss it a little bit later, is the organ of bacteria which help it to produce proteins. And for us, it's especially important because some parts of this ribosome is used for detection of certain pathogens and certain bacterial cells. It used to determine taxonomic composition of bacterial communities and to determine the amount of certain bacteria in the media. And the most important thing for us is nucleoid, is a place in bacteria where DNA is located. And all bacteria has the genome made up from DNA as the same as we are, but we are humans have 23 pairs of chromosomes, and these chromosomes are like plain. But bacteria have the genome structured in different way because they have only one circle chromosome, and it's called nucleoid. And this chromosome usually located inside the cell around the center of the cell, protected by cell membrane. This DNA contains all information about how bacteria will be structured, contains information about all proteins which bacteria will have, and these proteins is charged for every function of bacteria. So if we know the DNA structure, we know what kind of bacteria it is, and we know what it can do and what it cannot do. We should note that there are many different types of bacteria, and they have many different types of cell wall structures and different structures. For example, you may know gram-positive bacteria and gram-negative bacteria. They have different structures of cell wall, and the gram-positive bacteria have very like thick layer of peptidoglycan. Which is the polymer of peptide and partly peptide and partly glycan, like which helps to protect bacterial structures from outside dangers. And the gram-negative bacteria is more like progressive types. They have thin layer of peptidoglycan, but they have two layers of a lipid membrane, which help them to more successfully interact with outside, interact with environment, and a lot of pathogens has gram-negative structure of cell wall. But we should note that there are different ones, and if we want to extract DNA from bacteria, we should notice that we should try to extract DNA from bacteria, gram-positive bacteria and gram-negative bacteria with the same efficiency. So our methods should include these differences. Okay. Except the cell wall, we should discuss the ribosome structure. As I told previously, a ribosome is the organelle of bacterial cell. We have two, but our ribosomes have some different features. The ribosome in general involved in protein synthesis, and it has a very complex structure. In general, it combines from two subunits. One is large subunit and the second is small subunit. Usually in the cell, the subunits are free from each other and they combine together only when bacteria start to produce some protein. If we look closer to the ribosome, we can see that it consists of a lot of different molecules. The main parts of ribosomes is proteins and rRNA molecules. So here, RNA is ribonucleic acids, right? And as here, it serves as structural function along with the protein. So ribosome consists mainly from protein part, like S proteins, L proteins, and rRNA part. There are a few different rRNA molecules in ribosome. The most interesting for us is 16S rRNA molecule because the gene which is encoding this molecule is oftenly used to detect different types of bacteria. We will discuss this a little bit later. But in ribosome, we have a lot different, how do you say? We have a lot of different rRNA molecules, like 23S RNA molecules, 5S rRNA molecules, and a few more actually. These molecules differ from each other by size. Because this S after the number is like sedimentation index. This is the speed. Oh, I should turn off the commenting, right? This is the speed which which these rRNA molecules are sedimented in a centrifuge. So this is like speed of sedimentation of each molecule, which index their size. So the biggest molecule is 23S RNA, and the smallest 5S RNA, and the medium size has 16S RNA molecule. Okay, we will discuss about their function a little bit later. And we should discuss nucleoid, the most important thing, the most important organ of bacterial cell. You can see this is a photograph, like with microscopic micro photograph of bacteria, and you can obviously see the nucleoid inside the cell. It contains DNA, nucleic acid, which contains all information about all proteins and RNA molecules in this cell. A DNA sequence of this DNA is unique for every species of bacteria. So if we can read it, we can use DNA sequencing of often use certain regions, specific regions of DNA, to determine which genera or species of bacteria are present in our sample. If we have database which contains a lot of DNA sequencing of this bacteria, then we can read DNA of bacteria in our sample and compare to the database. So using these methods, we can know which bacteria are living in our sample and detect pathogens and any interested bacteria in any sample. Today we have a lot of like big databases with bacterial genomes, with bacterial 16S RNA genes. These methods of detection are very progressive and help us to describe almost any sample, except the soil, because in the soil there are a lot of uncultivated bacteria which are not described yet. So it's difficult to use these methods to describe soil bacterial community. But about the methods, we will talk a little bit later. Okay. What is the traditional methods of detection of bacteria? Traditional methods is a cultivation on selective media. So we have a petri dish, and after we have a sample, we try to grow bacteria which we have in the sample on some different medias. And as we know, different bacteria have different metabolism, so they require specific conditions to grow. We can try to uh, make these conditions in laboratory and try to grow different types of bacteria on a petri dish. We can use some nutrients, some different types of media, and if we have our sample, we can sow it on different media and see what is growing and what is not. That's how we usually describe microbiological structure of any sample, and it was the main methods to describe this microbiological structure for many years. Um, yes, but after we have grown or we grown all bacteria in our petri dish, we should describe what is their taxonomic structure, what is the taxonomy of our bacteria. Usually, it is done by morphological analysis. So we use some dyes, some special preparations, and look at these bacteria in the microscope and describe what we see. That's how we can differ different species of bacteria. Another parameters is cultural parameters. For example, it could be the type of dots, it could be the color of dots, it could be the form of culture. It could be a circle, a round form, or it could be some lines. It's different types of colonies helps us to describe species as well. And also, it's more like recent parameters, but it's already wide used. It's biochemical features. For example, we understand that this type of bacteria cannot produce this sugar. So we add this sugar to the media and see does this bacteria eat this sugar or not. If it's not, we know some about this bacteria, about their metabolism. So that's how we can describe bacteria using conventional methods on a petri dish. But these methods have certain limitations because it depends on cultivation, and as we know today, as most of bacteria species are uncultured, so we cannot cultivate them. And, the reasons could be different. Some bacteria needs to very specific and very complex conditions to grow. For example, they can grow only in a big community, only in a specific trophic chain and a specific microenvironment. We cannot make this microenvironment in the laboratory, so we cannot grow and we cannot describe that bacteria. Another limitation that growing of bacteria takes a lot of time. So it takes 2 to 5 days to grow a colony of bacteria, depending on the type, depending on the species. It could be in a different time. And one sample could contain from 100 to 200 different genera of bacteria. So if we try to describe the microbiological structure of the sample, of the one sample, using these methods, we have to do a lot of work, right? We have to sow a lot of petri dish and see what's happening. It's very difficult work. And moreover, we may have some dangerous organisms in these samples. So working with living pathogens or growing pathogens on a petri dish in special media is dangerous process. We can harm ourselves or harm other people using this. So it's dangerous and requires special conditions, special equipment, and special like professionals who work with pathogens, with living pathogens. And moreover, on top of that, morphological and biochemical analysis are inaccurate. So a lot of species of bacteria look very similar to each other. And their differences we cannot see using microscope, we cannot see using culture parameters or biochemical or general biochemical parameters. So it's very difficult to separate these species from each other using these parameters. So that's why conventional methods using cultivation are very limited, And now we have new improved like modern methods which help us to describe a lot of different bacteria in any type of sample. These methods do not depend on cultivation, so it helps us to not stuck on these limitations. Modern approaches using DNA of bacteria or any microorganism to describe its structure. It makes itself to work with pathogen because we don't necessarily have to work with living pathogens. We should extract DNA from the sample, and then DNA is usually harmless. It cannot infect any people. It cannot cause any sickness. So work with DNA is very safe approach. Moreover, DNA is very well stored, so we can store DNA in a freezer for a long time and work with it as many times as we can as we want to. And also these approaches using DNA has better accuracy in describing the kinds, in describing the genera or species of bacteria than classic methods. We should speak a little bit about DNA structure. As you know, DNA is a double-stranded spiral, and it consists of 4 types of nucleotides. It's adenine, guanine. Так, sorry. I thought I turned off the commenting. Wait a second. Please do not draw on the slides. I thought I turned it off, but maybe Nishesh, you should turn off the commenting so people cannot draw on slides. Okay, thank you. So there are 4 types of nucleotides inside the DNA: it's adenine, guanine, thymidine, and cytosine. They bind it to each other. And this is a code. This is a like genetic code which contains information about all peptides, about all proteins and RNA molecules. So it contains information about the metabolism of any cell, of any bacteria. The most often used for describing microbiological structure of samples is the gene of 16S rRNA. Of 16S rRNA. Why is that? We should understand that this rRNA molecule is a part of ribosome. So it is a part of very important structure in bacterial cell. And if here will occur any mutation, this mutation may disrupt the working of ribosome, and so if ribosome cannot produce their own proteins, the cell will die. So this is very, how do we say, conservative region. This region does not go with any mutation. So in the process of evolution, it changes very slowly. So we can know for almost sure that this 16S rRNA gene will be the same inside one species of bacteria and will differ a little bit in different genera and different classes. So using the structure, using the sequence of 16S rRNA region of 16S rRNA gene, we can separate one bacteria from another. And that's how we describe the taxonomic structure of our samples. We read this gene and compare the sequence of this gene with databases. The databases have a lot of already described 16S RNA genes, which annotated with the species of bacteria. So we can use this tool to describe our samples. Okay. Now we have 2 main molecular approaches which help us to describe bacterial communities in any types of samples. The first one is 16S RNA sequencing. It is commonly used for determination of microbial structure in the sample, and it gives us the list of bacteria. But we should understand this sequencing, it's a so-called high throughput sequencing or new generation sequencing. The method is very powerful because it's very sensitive. It help us to detect any tiny amount of bacteria in sample, but it is not quantitative. We cannot say how much of this bacteria we have in numbers. We only can say the ratio between different types of bacteria in our samples. This is powerful and useful as well, but sometimes we need quantitative methods. For example, if we work with pathogens, we always have to know how many cells of this pathogen we have at the sample, because we should understand how dangerous this sample for people, for animals, or for maybe agricultural plants. And we should understand how many pathogen load we have in the sample. For this purpose, we have to use quantitative methods to describe microbiological structure. And the most widely used quantitative methods of detection of specific bacteria is a real-time PCR reaction. We will speak about these methods today, later in the presentation. But now I want to say that what we want to discuss now is the limitation of the methods. This method is very precise. This method is very sensitive. I mean, real-time PCR reaction. But it helps us to, in one reaction, we can measure only one type of bacteria. So if we have to work with 200 types of bacteria in the sample, it is very difficult to perform these methods. So in our project, we will pick a special strategy to combine these methods and evaluate chosen type of bacteria very precisely. How we will do it? First of all, we will collect samples from any environment we choose, and then on these samples we will extract DNA, and this DNA firstly will go through 16S RNA sequencing here on Illumina MySec platform. After that, we will have a list of all bacteria and archaea which we have in this sample. We will analyze this list of bacteria and archaea and pick the most dangerous and the most interesting types of bacteria. After that, we will prepare real-time PCR reaction for each type of this bacteria, and we will know for sure how many this certain chosen, like bacteria of interest, we have in the sample. That information will help us not only describe general taxonomic microbiological structure of the sample, but it will help us to evaluate how dangerous the sample for animals or humans or plants, because we will know exactly how many pathogenic cells we have in our sample using the combination of these two methods. Okay, but before we go to these methods, these procedures, first of all, anyway, we should extract DNA, right? The first procedure that we will all do with any sample is DNA extraction. And this is and I think the most important one, because I think a lot of you will do it by yourself in your laboratory. Due to coronavirus restrictions, unfortunately, for now, I cannot go to your laboratory and help you to do that. But now we are in the progress of buying all necessary equipment to extract DNA and store it. And maybe later, a few months later, the coronavirus restriction will fall, and we will have a possibility to make offline educational seminar when we will pick some samples and then together extract DNA with them. Maybe I will do it myself first, and you will watch, and then you will do it by yourself, and you will have like actual skills to how to extract DNA from different types of samples. But now we will discuss theoretical basis of these methods. I will show you what equipment we have to use to extract DNA, and tell you how to do it in general. We discuss the general workflow and some details. Actually, we will go through detailed protocol of DNA extraction. So what the main points we should keep in mind when we extract DNA? First of all, we should extract as much DNA as possible from any sample to catch all microorganisms. Because if we want to adequately describe microbiological structure, we should catch all even minor fractions of microorganisms in our samples. So we should extract as much DNA as we can. The second point is DNA from different microorganisms should be extracted with the same effectiveness, because I mentioned in the beginning of the presentation that different types of bacteria, different types of cells have different structures of cell wall. So they have different like living forms. And to extract DNA, obviously, we have to destroy cell walls. And we have to pick the methods which will destroy effectively all cell walls, all different kinds of cell walls of bacteria to extract DNA from them. If we cannot destroy the cell wall, we cannot extract DNA. All other preparations will be useless. And the third point, DNA after extraction should be purified from all unnecessary chemicals, from all chemicals we use to extract it, and from all inner cell structures, because inside the cell, a lot of different molecules, proteins, lipids, and like some polysaccharides, a lot of different biochemical substances, molecules, which will which may sorry these substances should be purified, should be cleaned before we go to the following preparation, before we go to PCR or before we go to DNA sequencing. And we should purify our DNA, because these procedures are very sensitive, and they cannot go with some impurities in DNA. So how we do the extraction? On the slide demonstrates, you can see the photograph, and this cloudy thing in the tube is DNA itself. Yeah, it's look like glue or something. It's not so romantic as I imagine in school. But this molecule, this glue in tube, contains a lot of information about how living organisms are structured. And this is interesting and beautiful. Okay, how we extract DNA in general? General workflow is very simple. First of all, we have to process our sample. For example, if we extract DNA from soil, if we extract DNA from some sludge or from some plants, we should homogenize, right, our sample to make it, to destroy as many cells as we can. There are different methods to do this. As methods could be electrical, methods could be mechanical, and methods could be chemical. Usually in the protocol, we have some combinations of these three approaches, and we will use the combination of mechanical and chemical approaches to destroy the cell walls and destroy cells to extract DNA from them. After we destroy mechanically our cells of bacteria in the sample, we going through to the lysis. Lysis is a stage when we add to our sample some very aggressive molecules, which make some holes, make some like pores in the lipid membrane of bacteria, and it allows DNA to go outside. After that, we should remove all proteins from the mixture, from the solution. For this, we use a special reagent, and I will talk about them a little bit later. So this is a purification step, right? When we remove proteins and other different molecules. After that, we should purify our DNA, which we have in solution, even more. We don't have proteins, but we still have salts. We still have some inorganic compounds. We should wash it out. So for this purpose, we will use some special columns which can, in certain conditions, bind DNA and in different conditions release DNA. And how it will work? First of all, we will bind DNA on a special membrane in column. After that, we will wash it with a special solution which will wash salts and other impurities, but cannot wash DNA from the membrane. So DNA will be bind to membrane and will be washed by special solutions. And after that, is a final step is called elution. This is when we want to wash out our DNA from membrane because it's already clean, and we want to have a clean solution of DNA in our tube. That's the general workflow in the protocol of DNA extraction. Now let's go to discuss. Let's move to discuss a protocol which we will use especially for our application. For our application, we will use the Easy Power Soil Pro DNA extraction kit, and this is on the a photograph of it, and I have it my own here, and you will have the same box in your laboratory. It's already bought, and I think it will be shipped to your laboratory very soon. Why we choose this protocol? First of all, it's adapted to efficiently destroy the cells of all bacteria and fungi and almost all types of the samples. We can use soil, sludge, we can use different types of samples, and our DNA, the cells in that will be destroyed very effectively. You will understand later how it works. Now, compared to the alternative methods, it's up to 8-fold higher DNA yields because it has very efficient lasers. It has very high DNA yield compared to alternative methods. And this protocol includes 2 additional steps of purification. So we have recovery of inhibitor-free DNA for direct use in JS. It's a new generation sequencing of 16S RNA sequencing, or we can use it right immediately without any additional preparation. We can use DNA from this protocol to make PCR or sequencing. This is very comfortable methods. It's very suitable method for our purposes. And because of all these parameters, we have unbiased results, accurately represent sample alpha diversity. What does it mean? When we extract DNA, some protocols extract DNA from, for example, they extract DNA from gram-negative bacteria very efficiently, and but from gram-positive bacteria, not efficient. So after DNA extraction, we have shifted ratios between different types of bacteria in the sample. And after we do sequencing, we will have wrong numbers of bacteria, wrong ratios of bacteria in the sample. That's bad, and we want to avoid this situation. That's why we use this method, which helps us to have unshifted, unbiased results. It destroys all types of cells, it destroys all types of fungi and other types of cells. So it have very good results at the end. Okay, what we have inside this box. I will show you, and the slide demonstrates the list of what we have inside the box, inside the DNA extraction kit, and I will show you some tubes here. So the first is Power Beat Pro tubes. This is how it looks like. I will show you more closely. This tube contains very small beads, and this is a very important tube because this tube is devoted to the first step for the homogenization of the sample and for the destroying all cell walls, and it do it very effectively because what we do, we add the sample inside here and then shake the tube very fast on the special shaker. These beads it's like small stones actually, which do like this and destroy all cells. And moreover, the solution here. It contains a lot of strong chemicals which makes pores and in bacterial membranes, and so it allows all DNA go out. All DNA go out of the cell and inside this first step using power bead proteus. What else we have? We have special spin columns which will bind our DNA for purification. That's how it looks. I will show you. That's the column, and we add our DNA solution here, and then push it through the membrane. Here is a membrane, and DNA is bind to the membrane, and then we can wash it with different solutions to make it very pure and clean. So the solutions CD1, CD2, and 3, and so on, is the solutions contain special reagents for the lysis, for the purification, for the washing of DNA, solution which help to bind DNA on membrane, and we will add it to DNA one by one in the order from 1 to 2 and 3 and 4 to make all preparations. And we will collect our DNA in a special microcentrifuge tubes. It looks like standard Eppendorf tubes. It looks like that. Now, all of this we have in the box. All of this we have in our DNA extraction kit, like that. And you cannot buy any additional thing. I will tell later what you have to buy additionally, and we will provide all this equipment as well. So what about the storage? Because this box with the DNA extraction kit will be shipped to your laboratory, I think earlier, before we can start to work. And you should store these reagents very properly to make it work, right? So about the storage, that solution CD2 should be stored at from 2 to 8 degrees Celsius. So it should be, the better way is keep it in the freezer, in a fridge, not in a freezer, in a fridge on +4 Celsius temperature. And all other components and reagents of this kit can be stored at room temperature. You can see the parameters here on the slide. It's very comfortable and convenient for us that a lot of components of this protocol, a lot of solutions can be stored at room temperature. Because, for example, in PCR, in what's going following that, yes, in PCR, a lot of reagents for PCR reaction should be stored, some on the freezer at -20, some at +4 in the fridge, and some at room temperature. So it's very, it's not easy, not an easy task to store it properly. But here, this protocol is very easy, and it helps us to store it on room temperature mostly. Okay, this is the main procedure of our DNeasy Power Soil Pro Kit. And you can see the same scheme as we discussed previously. First, the preparation of sample. We add sample to this power bead tube and add solution CD1, which contains aggressive chemicals to destroy the cells. And then we should perform cell lysis. We will attach these tubes to the vortex adapter and place it into special homogenizer. Place it into some shaker, which can shake it for like 10 minutes or 15 minutes, and then mix it very thoroughly to destroy our cells. Then we start to purify. We should sediment all chemicals from the solution except our DNA. And we add solution CD2, which has a special reagent which can help sediment all proteins and other inhibitors of PCR reactions. Then we go to binding DNA. We add solution CD3 and load this to the spin column. So after we add in CD3, it creates a special conditions. It creates special conditions which allows DNA bind to membrane of column, and all other things will go through column, and we can discharge the throughput, this water, and save the column which contains all our DNA. After that, we wash it 2 times with different solutions, with solutions EA and solutions C5. These solutions have an additional purification steps because it wash out all salts, unnecessary salts, all inorganic compounds and other possible inhibitors. And then after we have very pure DNA on the membrane, we can DNA from membrane, so we can wash it out to the storing tube, collection tube. For this dilution step, we actually can use just water. We can use just very clean distilled water because this water have such conditions. It creates such conditions for DNA, so it's unbind from the membrane and can be washed out. Okay, this is a general workflow, and let's go to discuss it in details. First of all, we should think what equipment and reagents we have to have in our laboratory, except these balls, right? What we have to buy additionally. First of all, it should be microcentrifuge. It should be a Eppendorf microcentrifuge, which have speed up to 16,000 G. We should have pipettes like this ones. I have my own in lab. For these preparations, we have to buy 2 types of pipettes, and we will provide them, of course. One for volume from 100 to 1,000 microliters, and another, the smaller one, from 20 to 200 microliters. We have to use these 2 types of pipettes. For homogenization, for lysis step, we should use Vortex Genie2. I cannot show you it because my Vortex in laboratory room and I cannot move it to here, but we will provide this Vortex as well, and the Vortex adapter to place on them all our tubes, all our lysis tubes, power bead tubes to destroy cells in samples. Okay, let's go through protocol. What's important to know before starting protocol? We should be ensured that the power bead tubes rotate freely in centrifuge without rubbing, yes? And if solution ZD3 is precipitated, some solutions which will be shipped to you in the box may precipitate because of the wrong storage conditions. To avoid precipitation, solution CD3 before we start to work could be heated up to the 60 degrees Celsius and shaked until precipitate dissolved. Only after that it should be cooled down to room temperature slowly and could be used in procedure. And all procedures, all steps of this protocol should be performed at room temperature. We should be ensured, we have to be sure that all our reagents have room temperature because they work properly only at room temperature. If they are cold or heated, the protocol could go wrong. The first step of procedure is the spin power bead pro tube briefly to ensure the beads have settled to the bottom. This means that we should just shake this and ensure that all beads, all these little stones, are seated on the bottom. Then we should add up to 25 milligrams of soil or different types of our sample and add solution CD1 to start lysis. Then the vortex briefly to mix. After sample has been loaded into the power bead tube, the next step is a homogenization and lysis procedure. So the power bead pro tube contains a buffer that will help to disperse all soil particles and disperse particles of our sample. And then to begin to dissolve humic acids and protect nucleic acids from degradation. This vortex step mix the components in the power bead tube and begins to disperse the sample in the buffer. Next step is homogenization, of course. For homogenization, we will use Vortex Genie2 and the special adapter. Unfortunately, I cannot show you this vortex, but you will have it in laboratory, and it's very simple equipment. I think you will figure it out. But anyway, I hope I will have a possibility to come to your country and to make a special training, which and show you how to work with any equipment inside this protocol. What we should note that using vortex adapter for more than 12 preps, but I think we will use for 24 or 12. I don't remember exactly. We should increase vortex in time, so we should vortex 10 minutes more. And using the vortex adapter, it will maximize homogenization, which can lead to higher DNA yields. We should avoid using tape, which can become loose. So for the signing of every sample, we should use a special marker. I can show you. It should be a permanent marker like that to sign any tube. And we should not use any tape because it could be loose and wiped out during the homogenization. Vortexing and shaking is critical for completing homogenization and cell lysis. Cells are lysed by a combination of chemical agents from step 1, you remember CD1 mixture, and mechanical shaking introduced at this step. Randomly shaking the beads in the presence of disruption agents will cause the beads to collide, and then will collide with microbial cells and lead to the cells breaking open, and DNA will go out to the solution from the cells. Okay, let's go next. Next is the centrifugation of the power bead per tube, because our DNA is already in solution, but we should get rid of any broken cell parts. So we will centrifuge our tube at 15,000 G for 1 minute, and all broken cells and cell structures will be sedimented, will be pushed to the bottom, but our DNA will stay in the solution. And then we will transfer the supernatant, so the solution without any particles, to a clean 2 milliliter centrifuge tube like this. This is provided for this kit. We should expect from 500 to 600 microliters of the solution. I usually take 500 microliters, and you should pick the number which you will try to pipette. You should note that the supernatant may still contain some cell particles. On this step, containing some small amount of cell particles or some particles in the sample is okay, because then we will have purification steps which will help us to get rid of them. On the 5th step, we will add 200 microliters of solution CD2 and vortex to shake it, just mix it. A solution CD2 contains inhibitor removal technology, so it's a special combination of reagents which get rid of any possible PCR inhibitors or sequencing inhibitors. This is the substances which we don't want in our solution, and this is a reagent that can precipitate non-DNA organic and inorganic material, including humic acids, cell debris, and proteins. It's important to remove contaminating organic and inorganic matter that may reduce DNA purity and inhibit downstream DNA implications. After that, we mix that CD2 solution with our DNA solution. We centrifuge it at 1500 G for 1 minute to precipitate, to push to down all the impurities. There will be a pellet, a special, like, I don't know how to say, you will see a pellet, this is white pellet on the bottom, which contains some all impurities, proteins, cell structures, and everything. After that, we should take out from 500 to 600 microliters of DNA solution, and we should try to avoid this pellet, avoid these impurities as much as possible. They should stay in the provided microcentrifuge tube. And then we move our clean solution to another tube. We should note that the pellet at this point contains non-DNA, organic and inorganic material, including humic acid and, as I already told, cell debris and proteins. Yes, we should really avoid transferring any of the pellet in the next tube, because we want to get rid of this. The next step is the solution CD3. This solution is a high concentrated salt solution. It creates the special conditions which can help DNA to bind to the membrane of our column very effectively. So this is a high concentration salt. We add and shake it, vortex it for 5 seconds to mix with the solution, and this will adjust the DNA solution to allow binding of DNA, but not non-DNA organic and inorganic materials. So this material, which is not DNA, will not bind in these conditions to our membrane. So it goes through, and we can discard all liquid which goes through the column. We will load 650 microliters of the lysate containing CD3, of course, into spin column. I already showed you a column. I will show again. Into these columns, we open it and load here with a pipette, like here, 650 microliters of the lysate. And after that, we'll centrifuge it at 15,000 G for 1 minute. It will push the liquid part through the membrane, and DNA can selectively bound to the silica in the membrane in the column in the presence of high salt solution. Contaminations will pass through the filter membrane, leaving only DNA bound to the membrane. Then we will discard the flow through, so we discard the liquid which pushed through the membrane, and repeat this step, because we have much more lysate. We have after all previous preparations, we have about 2 milliliters of the solution. We can only load for one time. We can only load 650 microliters. So we should repeat this step 3 times with one membrane to load all DNA to the column. After that, we should place this column into new collection tube. It should be clean and it's provided. Avoid any splashing of any material of previous solution, because it contains a lot of impurities. So after that, we add 500 microliters of solution EA to the column and centrifuge it at 15,000 G for 1 minute. The solution EA is a wash buffer that removes protein and other non-aqueous contaminants from spin column filter membrane. So this is the additional washing step. The conditions on the membrane is so that we can wash it with this solution EA, and the DNA still will be binded to the membrane, but impurities will be washed out. Then we discard the flow through as always, and then add solution C5 to the last purification. Solution C5 is an ethanol-based wash solution used for further clean of DNA that is bound to the silica filter membrane and the spin column. This wash solution removes residual salt, which is very important, humic acid and other contaminants, while allowing the DNA to stay bound to the silica and the membrane. For the last time, we are discarding the flow through, discarding the C5, which pushed through membrane, and go to the next step. After that, we should spin the empty column without any solution to dry it from C5, because a small amount of solution C5 is resting on the membrane, and we should push it out. So we are spinning it at the maximum speed for 2 minutes, because this removes residual solution C5. It's critical to remove all traces of solution C5, because the ethanol in it can interfere with the downstream DNA application. Ethanol will break our PCR reaction, will stop DNA sequencing, so we should get rid of that before we elute our DNA. And then we go into the last step of our protocol. We add solution C6, about 50 to 100 microliters, and I prefer to add 100 microliters because it gives us bigger volume of DNA in the end. We should add it carefully to the center of the filter membrane to make sure that we have all membrane wet with solution C6. Placing solution C6 in the center in the small white membrane will make sure that the entire membrane is wet. This will result in a more efficient and complete release of the DNA from the column, and solution C6 pass through the silica membrane. DNA that was bound in the presence of high salt is released by this solution. Actually, is a so-called trace buffer, but the just distilled water will works as well. And after that, we centrifuge for the last time at 15,000 G for 1 minute. And we can discard, we can throw out the column because it's empty, and all our DNA will be ready for downstream application. And we will have a tube with DNA which contains very, very pure solution without any proteins, cell structures, contain only pure DNA, and it will be total DNA. It will be total DNA from our sample. I think I can show you the tube like this. I will have it in my fridge. Have 1 second, I will show you. And that's how it usually looks like. It's a small tube. It's freezed, and it contains freeze DNA. What here you can see it. You see it clean. It's transparent, so it has no impurities. And now it's ice. And I keep it in a freezer as it's recommended at -30 degrees Celsius. So it helps to protect DNA from degradation for a long, long time. It could be stored at -30 degrees Celsius about 2 years, 3 years, 5 years, and this DNA will be okay, and we can use it for analysis. I will take it back to the freezer. Okay, so that was a protocol for DNA extraction. I hope it was informative. And last thing I want to tell you about this: do not forget to sign all tubes before you start. So we will talk about it offline on offline seminars later, but we should remember always that before we start to work, we should know how we will sign any tube. How we will sign any tube in our sample. So we should know the name of any tube and sign all tubes for the preparation before we start. That's how we make it like we will not be confused in the end and know this DNA belongs to this sample, and this DNA belongs to different sample. It's very important because you cannot see on DNA from which sample it is. So do not forget to sign all your tubes. Okay. So after we extract all DNA, we should measure the concentration of DNA. For this purpose, we will provide to all laboratories a qubit 4 fluorimeter. Today, we're not going through detailed procedure of DNA concentration measurement. It's easy. It's very common procedure, not so hard to understand. But we don't have so much time to discuss it. This machine use fluorescent dyes. So I describe you the principle. The DNA, we cannot see the DNA in the tube, but now we can add a special dye which binds to DNA. And this dye, when it binds to DNA, is starting to glow. And this glowing we can measure with fluorimeter. And this fluorimeter can measure the amount of glowing of the dye combined to DNA, and that's how this measures the DNA concentration. As this procedure, we always also will cover in our offline seminars later in the project. And after that, I think today we have the time for to discuss a little bit about polymerase chain reaction, because it's very important and powerful methods. And it's interesting that this is a this is a nature-based process. This process is based on the actual DNA duplication, DNA replication in a living cell. And let's remember how the living cell duplicates in general. Duplication of bacterial cell is called simple binary fission, and it have not so many steps. Now, on the first steps, like in human cell, dividing bacterium needs to copy its DNA, right? Unlike human cells, which have multiple linear chromosomes, bacterial cells usually have one single circular chromosomes, this should be duplicated before the cell will be divided for two. Copying of DNA by the replication enzymes begins at a spot on the chromosome, purple here, is called the origin of replication. This spot is called the origin, and this is the spot in bacteria where duplication started. As replication continues, the two origins move towards the opposite the ends of the cell, pulling the rest of the chromosome along with them. And the cell also gets longer, adding to the separation in the newly forming chromosome. I will show you later. You can see here the cell elongates, becomes longer, and all origins go in different directions from each other. Then replication continues until the entire chromosome is copied, like here, and the replication enzymes meet at the far sides here. Once the new chromosomes have moved to the opposite cell's ends and clear the center of the cell, division of the cytoplasm can take place. In this process, the membrane pinched inward, and a septum or a new dividing wall, here we can see a septum, forms down in the middle of the cell. Bacteria have a cell wall, so they must regenerate this wall when they undergo cell division. So they have to build the wall here in the middle of two newly dividing cells. And finally, the septum itself splits down in the middle, and the two cells are released to continue their lives as individual bacteria. That's how, in general, division of bacteria is going, and we should look at more closer. We should look. I will show you. We should look here, very close. What we can see here, we have double-stranded original DNA sequence, right? To duplicate that, we should firstly separate two strands from each other. We have a special protein for that, helicase it's called, and then build up a new strand on each original strand. You can see how it's done. Now, the first strand is leading strand because the direction of polymerization is going along with replication. But we have lagging strand because here direction is the opposite, and then the processes of DNA synthesis here and here a little bit different from each other. But that's how it performed in a living cell. In general, it's very simple because we have the protein DNA polymerase, which use the original template DNA. Like it uses it as a template and then binds the copy of it using the template as a source of information. What is important in this for us, for PCR reaction? That this DNA polymerase cannot use just template, cannot use any template to start to duplicate it. They have to use a primer. They have to use short sequence of DNA which can bind to template and then serve as a primer, serve as a start for new strand DNA synthesis. This primer binds to specific place on template DNA, not any place. This place is located in origin on bacterial cells. So it cannot be bind to any place on DNA. And that is a very, very important feature which we are using in PCR reaction. In general, what is PCR? PCR is a biochemical process capable of amplifying a single DNA molecule into millions of copies in a short time. This amplification is achieved by a series of 3 steps. The first step is these steps actually copy bind to flanking regions of target DNA. So here, when primers binding to templates. And then the third, we have extension. When DNA polymerase, the protein which synthesizes DNA, from 25 to 35 times to exponentially produce exact copies of target DNA. And you can see how it looks on the slide. On the first cycle, we see it in details. First, denaturation of copy of the template DNA, annealing of primers. You can see we have pair of primers, and then extension. Here we have DNA polymerase, which extend this new copy of DNA. Then we move it one more time, the second cycle, third cycle, and this 30th cycle. We have two very like huge amount of copies. You can see how big, how huge numbers we have here. So using these methods, we can detect any bacteria inside our sample, because we can specifically amplify the DNA of any bacteria we choose using the specific primers. This is the secret of the specificity of polymerase chain reaction, because we can design primers which bind only to specific bacteria. So we add to the PCR reaction our specific primers. And if reaction goes, we can say this bacteria occurring in our sample is present in sample. If reaction doesn't go, so primers do not bind to DNA, we know that this bacteria is absent. And that's how we use PCR for detection of microorganisms. We design a special primers which can specifically bind only to DNA of specific bacteria. And we have a lot of tools for designing these primers. For example, Primer Blast is a very powerful tool. It has certain limitations, but it's okay in general. And we can design the primers by ourselves, but it's very important and difficult task to design primers for every bacteria we want to analyze. So we will use literature. We will use some published data on primers, which primers are good with different types of bacteria. And for quantitative analysis, to quantify how much bacteria we had in our sample, we will use real-time PCR reaction. It is a modification of PCR which help us to quantify the results. How it works? We should add to reaction special dyes. And this dye have a special, how do you say? This dye have a special feature that when it's free, it's unbound to DNA. It does not glow. It's fluorescent dye. But when it's bound to double-stranded DNA, it starts to glow. So the more double-stranded DNA we have in our reaction, the more glowing we have. So we can use this, we can quantify using fluorimeter, using special detectors, how much glowing we have, and then quantify how much DNA we have. And if we know how much DNA we have in the end, we can calculate how much DNA we had before. And so we can calculate how many cells of a certain organism we had in our sample. And then we can measure pathogenic cells, different types of cells, and evaluate the danger of these samples. Okay, this is another type of This is another type of detection system. This is called cyber green system. This is a TACMAN system. It works in different manner because this is the opposite mechanism. Here we have another fluorescent dye, reported dye, but this is does not glow when it binds to DNA. But in the process of DNA synthesis, this short sequence is destroyed and it releases dye. And we can calculate the amount of released dye in solution and then understand how many DNA we have. This is the same principle of detection, but different types of fluorescent dyes. Okay, and in the end, we have graphs, we have pictures like this. When on the x-axis we have number of cycles. On the y-axis we have the signal from the dye. We have the parameter which help us to calculate DNA concentration. And we can see that here's the threshold line. If our line will not go upper the threshold, we call the sample negative, so the reaction does not go. If it goes upper, we can remember on what cycle it goes upper, and this on line will help us to calculate the concentration of DNA. So this is how the PCR and real-time PCR is working in a nutshell. Of course, it's very complicated procedure. It involves a lot of different reagents, and we cannot discuss all these reagents here. This reaction, this stage of these preparations will be performed here in Moscow in my laboratory after we will extract DNA from all the samples in your university. And I think it's enough information for today. That's a lot of information for today here. And I want to thank you for your attention. That's it for presentation for today. If in case you have any questions, you can ask them now, because maybe I said something not really like maybe you didn't hear something or anything. Please ask now. Anyway, if you don't have questions, I will share my presentation with all of you. I will share the recording of this meeting, so you can learn it later. You can think of it later to see the presentation, and then maybe ask questions. You can, for asking questions, you can write a message on email to me. We will share email of me as well, and or any member of our team, we will share the list of the members, so you can write them down the message to ask any questions. Yeah, of course, I will show the presentation. Yes, Professor Khan, of course. Okay. So that's it. If you don't have any questions, I have 1 minute more for the questions. You can just unmute yourself. I think you can just unmute yourself and ask you want to ask.
Okay.
Yes, Professor Kitut.
Yeah. Okay, I'm the first or the students? Okay. Yeah.
No.
Okay. Thank you, Sergey. You have a very interesting presentation, and you are also a very great offer. Yeah, about we all can get your presentation PowerPoint and
Yeah,
and
of course.
also make a question to the next step. So actually I want to know, I have actually 5 questions.
Oh.
I write it here, but not so complicated, I think, because I don't so understand about this, about analysis. But I want to know about first, what part is the most difficult when doing this DNA extraction, especially using the NAC kit, for example, or other part. That is one, and the other is in the PCR analysis. When you're running a PCR, is it lagging strength will be duplicated?
Okay, thank you.
Yeah, that's it.
I will tell you.
Yeah.
The first question about the DNA
Yeah.
extraction. The most important and the most, I think, difficult part, because all preparations in DNA extractions are not difficult. It's just moving one solution to one tube and another solution to another tube. It's not difficult, but it is very important step that when we collect samples, we should make DNA extraction as fast as possible, to not wait for 1 day or 2 days, because bacteria start to change after we take out the sample, and we will have different results. If we wait after we take samples, wait and extract DNA, we will have bad results. So the most important in extraction is, after collecting samples, we should extract DNA as soon as possible. This is difficult because maybe it should be shipped to the laboratory, should be kept somewhere. This difficult one. But the process of extraction DNA not difficult. And the second question about PCR, yes, there is no lagging strength in PCR reaction, because in the living cells, they have how do you say, special protein in living cells separate two strands of DNA in very small region. It separate and then how do you say, not all DNA is separated in living cell, but it's separate and synthesis after that. But in PCR reaction, we heat the solution, and all DNA is separated. So there is only single stranded, one stranded DNA with one strand. All strands are leading strands.
Okay.
Okay. Thank you, Kitut.
Yeah. Thank you. Thank you.
Yes. We should.
Okay, sorry.
Oh,
Yeah.
I can see you.
Yeah.
Yes.
Yeah. Hello. Thank you for the presentation. And I just concerned about how we apply this content to the water treatment or the environmental technology, refer to the biological methods. So I hope that in next seminar, you will focus on this content, or from this, after we we extract the DNA, or we have a detailed research about some microorganism. So how we can apply this to the waste treatment. In our colleagues, in our faculty, my colleagues also extract some or isolate some specific microorganism to use this in to make the to cultivate, to make the biomass. Then they use for wastewater treatment and also solid waste treatment.
Yeah, it's thank you.
Yeah. So can you explain a little bit more? Yeah.
Yeah. So this is thank you for your comment, because these methods will actually help us to do something like you proposed. And but before we do that, because this project is educational project, which were devoted to sharing the methods. Yes, we want to teach students how to use methods. extraction of DNA, PCR, and everything, like how to work with results of these methods. And then we, after we can use it, we should, in parallel, think of what project we can create using these methods in like your local university. Of course, we want to discuss this. I think we will do the seminar about this as well, but a little bit later, after I finish with methods, the next seminar will be devoted to sequencing, to describe taxonomical structure. After I tell about all methods, we will make seminar about application, about how to in real life, what we can do with them, because it's important, of course, as well. Thank you. Any more questions? Okay, so if no questions, no problem, because we will have presentation and the recording, and do not hesitate to ask questions in email, and we will share the email and maybe WhatsApp as well, and ask any questions you will have. So for now, thank you very much to all people for attention.
May I ask a question? A little question?
Okay.
I want to know what the price of the DNASA kit for 50, for example.
It's about 700 euros.
7?
Yeah, 700, I think so.
700 what?
700 euros.
Oh, euros. Okay.
Yeah.
Okay. Yeah,
it's about like that. But we already bought that. We have a project devoted to this, and using money from this project, we already bought these kits, and they will be shipped to your university later. So you don't have to buy this, and you don't have to buy any equipment. We do it by ourselves and provide all equipment and reagents, of course, because that's some of them are very expensive. Now, for example, fluorimeter for measurement of DNA concentration is very expensive, but so we will buy it, of course, as well.
Sergey, this is I think this is a equipment cost, right? And how about the running cost? Or how much we have to spend for one test? You have this information?
Oh, actually, I don't know exactly for now, because it depends on different parameters. On one test, not so much. Actually, PCR tests are very cheap. PCR test equipment for PCR is expensive, but tests are very cheap. And I in the laboratory already have equipment, so we will make tests here. Yes. So, and for now, we cannot calculate the exact number because we have to know all expenses about equipment. Sorry.
Okay. Yeah. Thank you.
Yeah. When will you send the PowerPoint? Your PowerPoint?
Send what?
Yeah.
PowerPoint.
Your PowerPoint.
Today.
Okay.
Right after we
Thank
finish,
you.
I will send. I think not me. It will be. I believe it will be Natalia or Natalia. It will be. I will inform you later about the date of the next presentation. And with Yuki Tut, we'll have an offline presentation maybe very soon.
Yeah. Okay.
So goodbye, everyone. Thank you for attention once again, and see you next time.
Yeah. Bye-bye. Thank you, Sergey. Thank you. Bye-bye.
Bye-bye.
Xin chào.
Xin chào.
Thank you.